Okay, so building on what we learned last time, we're going to do another form of metabolic study. It's the oldest form, and it's the easiest one to use. It's called sealed jar respirometry. Basically what we do is we take a jar, as the name would imply, and we put a fish inside of that, we seal it up, we let it sit a little bit, and then we measure the oxygen content that was used. So it's a really simple study. It's one that we use all the time when we go to places where it's really remote and you don't have access to electricity, for example, or field generators. Then you need to be able to somehow collect those types of metabolic data. It's really simple. You collect up the fish, you put them in either a glass jar, or maybe you've got a little respirometer that you've built and you've taken with you of a known volume. You measure the oxygen content in the water when you start, and again when you end, and the difference between those two then becomes your oxygen uptake. And of course you keep track of the time in between, because you don't want to, you want to express that in milligrams absorbed by the fish per minute or per hour, whatever your units are. Okay, so what we've got here is a setup where we have two tubs that are filled with water. One of them we're going to heat up 2 or 3 degrees, maybe 4 or 5 degrees warmer than the other. The other ones, right now they're at room temperature, so we'll have one at room temperature, we'll have one room temperature plus 5 degrees. And in that regard, it mimics what we did last week when you calculated the Q10 value. So when we calculate the Q10 value, then we'll be able to compare goldfish to other types of fishes that have been tested in this way. And it's pretty straightforward procedure for the most part. So I think you're going to like that. And again, we can use it just about anywhere. Okay, so what we're going to do is we've got goldfish over here in the tubs. And you can see they're swimming around. We're going to go ahead and take our take the goldfish out, and we will place them one at a time into one of these Erlenmeyer flasks. The Erlenmeyer flasks hold 500 milliliters of water, so we know there's half a liter of water in there. We're going to measure the oxygen using a Yellow Springs instrument or YSI oxygen meter. These are older style meters, but we use them a lot in the field. And again, they run off a battery, so you don't need electricity and a plug-in to be able to use them. What we're going to do first is go ahead and calibrate it. So they have an internal calibration, so we're going to go ahead and ask us for the altitude. We're at sea level here in Florida, so we can go to the next. Okay, and then it says 100, I didn't mean to do that. Let's try that again. There we go, altitude. Okay, and right now it's telling us that this probe, this is the probe itself, and this is inside of a area that's got 100% humidity in it, okay? And so right now it reads roughly 97% saturation. What we're going to do then is we're going to go ahead and set this so that instead of measuring it in percentage oxygen, we want it to give us the number in milligrams per liter. And oops, now it's asking me about salinity. I don't need that. And hit that one more time. And there we go. So 100% air saturation, then we've got 8.72 mgs per liter. Now, once we get it in the water, there'll be less than that because there's more oxygen in air than there is dissolved in water. Okay, so we can take that YSI probe and we can put that into the water and swirl that around. It does a couple of things. It gets our temperature, gives us the temperature, and then it'll also tell us what the oxygen concentration is in water in milligrams per liter. And so I'm going to go ahead and assume that all of the tanks are the same, well, the room temp tanks, all of these Erlenmeyer flasks are all identical. Okay, so I can just take the temperature and the reading in the water. And you notice that we swirl that around as we go along because this is what we call a Clark electrode. And in the process of measuring oxygen, it consumes it. And so if you don't keep moving the probe, you develop a dead space on that sensor, and you see your oxygen concentration go down and down and down. And so typically what we do is we have some system in place where we can swirl it around a little bit and keep renewing the surface on the cathode. So I won't go into the details of how Clark electrodes work, but if you're interested, you can look that up. And it's a relatively simple system. You can build your own, as a matter of fact. And I remember as a beginning undergraduate, we had to build our own oxygen meters using voltmeters and mercury and all kinds of bad things that we wouldn't let you work with now. Okay, so you can see we've kind of stabilized there. We're running at about 5.3 or so oxygen level. And we'll go ahead, and that's going to be our starting number for room air. So we need to go ahead and write that down somewhere. Just like that. So let me go ahead and we'll tell you what. Let you scroll that around. And so this is in the room temperature.
Mm-hmm.
So there's room temp. Okay, and so initial. So what do you remember there?
About 5.32.
Okay, and then we will have a final O2 at some point here. And we'll also have a room temp plus, how many degrees do you want to have on that? 5?
I set it to 28, so that's what it's sitting at right now.
Room temp was
22.3.
42 degrees, and heated is at, one more time, sorry?
28.
28. Okay, so you guys are going to need these numbers because you can calculate Q10, so that's the nice thing about that. We can measure metabolic sensitivity in these animals by doing the fairly simple equation that we did last time, so that's what we're at. Okay, so again, on the heated group, we'll also have an initial oxygen in the jar. And we'll have a 2 and a final. Go to okay, I think we got all the stuff that we need. All right, so the next thing that we're going to do then is we'll go ahead and we have aerated this water for an hour or more. So everything's saturated. We're going to go ahead and plunk the fish into the reservoirs here, into the Romine class, and we're going to put cork on them so that there's no air trapped, and we're going to try not to trap any air in it. Sometimes you get little bubbles, but overall we try really hard to keep the bubbles out. And we will let them sit for a period of time. And I think we're going to do them once every 10 minutes.
Sure.
Is that going to work for us, I think? So we'll put in one fish each into the room temp and into the room temp and into the heated. Then we'll wait 10 minutes and we'll do the next one, and we'll wait 10 minutes and do the next one. By the time we get to the end, we should be able to pull out the first one and measure the oxygen concentration. Okay, so we should move through it fairly quickly. I haven't done one of these in this particular fashion, so maybe we screw it all up, but we're going to give it a try and see. I'm going to assure you that no goldfish will be damaged in the process, so that's how we're going to go about it. Okay, with that in mind then, let's go ahead and we'll need to have a stopwatch. So I'm going to... Okay, so we now have our first goldfish in the room temperature tank and in the heated tank. And you can see that we've put the stoppers in and the goldfish are moving around. They're going to sit there for 10 minutes, and then we're going to go ahead and add the second goldfish in, and then finally the third. And at the end of a 30-minute period, we will measure the oxygen in the goldfish vessels that were set up first, and that'll give us our final reading. So we've got the initial readings on the board, we will put the final readings on the board, you'll subtract one from the other, and that'll give you the oxygen uptake. And you can also utilize, because we've got two different temperatures, you can also utilize the Q10 formula to calculate a temperature quotient. So once we get the next few fish in, we'll come back and take a look at that. Okay, so we have all the fish now in the jars. They've gone in at 10-minute intervals, and in just a moment here, we're going to take out the first fish that we put in both at room temp and at a heated bath. And we will use our YSI oxygen meter then to go ahead and calculate oxygen concentration. And once we're done with the oxygen concentration, we'll move the fish over, we'll take its weight and its length, and we usually keep track of those kinds of things. Sometimes that's important to the study. It doesn't affect the oxygen uptake at all, but we just want to be able to make sure that we can get a weight, what we call a weight-specific metabolic rate. So when you calculate metabolic rates this time around, what you're going to do is divide through by the mass of the fish. So we will collect the mass and length of the fish as we go through this experiment. Okay, so now we are going to go ahead and weigh and measure the fish. And so that's always a little bit tricky, but we're going to try to keep them in the net so that they don't get damaged. And we'll have standard length and mass in grams, standard length in centimeters. Okay, so this was fish number 1 in the room temp. Straighten up here for us a minute. This one is 3 centimeters even. And weighs 0.8 grams. 3 centimeters, 0.8 grams. You write that down.
Just write it on the board real quick. Left column.
What's that?
Left. Yeah, you're right. 8 gram, 3 centimeter.
Centimeters. One 1.8 grams.
Mm-hmm. Yeah, I believe so.
Okay, and so we're going to do this, well, 4 more times, and we will get the data sheet all set up, and then we'll talk about that a little bit. Okay, and so here's the data that we collected on all of the fish that we had in warm and in cooler water. You have access then to all the data on the board. We'll send out a sheet with that so that you'll have it. What I'll need for you to do is go ahead and calculate the average metabolic rate. We did that last time, that wasn't a problem. Also calculate the Q10, because we did have a warmer and a cooler group, and we'll see how that turns out for us. The other thing that I would like you to do is calculate a third value, which would be the total metabolic rate divided by mass. We call it the mass-specific value. That mass-specific value plays a big role in how animals grow and scaling. And so we'll talk about that later, or actually you will see the lecture on it, I guess, on the PowerPoint slides. But it's a really fascinating thing that you can get total oxygen uptake rate on something big you would expect it to be a really high oxygen uptake. But if you take that and divide through by mass, that number drops way, way down. Whereas if you're a smaller animal, you have a higher, much higher mass-specific metabolic rate than if you were a larger animal. So it's a weird thing. I want you to think about that a little bit. And then the worksheet will come, and I think that'll be a question that we'll probably put on the bottom. Why do you think that is? And I'll be most curious to hear your answers. And until next time then,